Assalamu alaikum warahmatullahi ta'ala barakatuh. Today we are going to start a new course, and that is amino acid metabolism. The content of this course include catabolism. We will be discussing how the body catabolizes the amino acid, particularly the carbon skeletal part of the amino acid and the amino acid nitrogen. And we are going to talk about some anabolic reactions and pathways, including synthesis of non-essential amino acids and of specialized products that can be synthesized from amino acid. And of course, as we go on through the course, we will come across some abnormalities that are related to amino acid metabolism. But today's lecture, our objectives include defining protein turnover, discussing what is meant by protein degradation, defining nitrogen balance and listing the types of nitrogen balance, to compare and contrast main types of protein energy malnutrition, to discuss how the body removes ammonia, and to discuss the role of the enzymes transaminases, discuss reactions of urea cycle, and to list the disorders of urea cycle. Amino acids are either, of course, supplied in the diet as essential amino acids or synthesized in the body, and this includes the non-essential amino acids, of course, by transamination of metabolic intermediates. And you know this list from your basic course in amino acid chemistry. The nitrogen of amino acid is removed by transamination and deamination and excreted as urea, while the carbon skeleton has three phases, either oxidized in the citric acid cycle, or it can serve as a glucogenic substrate, of course, by passing through the citric acid cycle first, or it can be used to form ketone bodies, which can be, of course, for oxidized for energy metabolism. And this is a colorful representation regarding the overall picture of amino acid metabolism. So proteins come from the diet. We have a pool of amino acid. This is around 250 to 300 grams per day. There is a continuous input out from tissue proteins. So these amino acids can be used to build up tissue proteins, and tissue proteins can be broken down to yield these amino acids. The amino nitrogen, of course, as we said, the pool itself can be used to form non-protein nitrogen derivatives. And then by transamination, we can shift or we can exit from amino acid pool into a carbohydrate, for example, by gluconeogenesis. We can use glucogenic amino acid to form carbohydrate. The amino nitrogen and glutamate can be deaminated to ammonia to form urea, as we are going to see today. And amino acid can be, particularly the ketogenic amino acid, can be converted to acetyl-CoA, which can enter citric acid cycle to be oxidized completely as energy source, or it can be used to form ketone bodies. What do we mean by protein turnover? Protein turnover is a continuous process of degradation and resynthesis of all cellular proteins. A normal adult degrades about 1 to 2% of his or her total body protein daily, and the majority of the liberated amino acids are used for protein resynthesis. So our body protein, particularly in normal adult, 1 to 2% will be degraded daily, and the carbon skeleton will be used as intermediates for synthesis of intermediates, amino, the nitrogen of the amino acid will be used to form urea. 75 to 85% of the amino acid will be used for the synthesis of body protein. Excess amino acids are not stored. This is an important fact to remember. the body does not store excess amino acids. They will be either used for protein resynthesis to synthesize new proteins, or it is going to be rapidly degraded. And proteins have different half-lives, or T-half, and T-half, or a half-life of protein, is a time. So it is a time that it takes for half of the protein molecule in a given population to degrade, and of course it is measured in a unit time, in minutes, for example, hours or days. We have examples here of short-lived proteins such as regulatory proteins, signaling proteins, and transcription factors. They are considered to have short half-lives measured in minutes to hours. Cytosolic enzymes can have a short half-life or T-half of hours to days. Membrane proteins and extracellular proteins like albumin and antibodies can have a half-life of days to weeks. For example, we know that antibodies, for example, immunoglobulin, have a half-life of about 3 weeks. Protein degradation is affected by many factors, including the amino acid sequence itself. There is called what is called an NN rule, meaning that the N-terminus end of the amino acid, if it has certain amino acids there, this will influence the half-life of the protein. Post-translation modification, for example, ubiquitination, adding ubiquitin to the protein in a post-translation modification can distance this protein for degradation. The cellular location of the protein itself, for example, the membrane bound and the secreted proteins are more stable, so they have longer half-lives, while the cytosolic proteins generally have shorter half-lives. Environmental factors such as exotic stress, pH, and temperature can also influence the half-life of the protein. For example, exotic stress is going to increase the rate of protein degradation, so it will decrease its half-life. An important fact here to remember is that proteins that contain PEST rich region, which refers to proline, glutamate, serine, threonine, those proteins tend to have short half-lives and more rapidly degraded. Why? Because this sequence or this motif often serves as the recognition signal that marks the protein for degradation. So a protein that has this sequence, it will be ubiquitinated, and it will be destined for proteasomal degradation. And there are two pathways for protein degradation, either ATP independent or ATP dependent. The ATP independent occurs in the lysosome mainly for membrane and associated proteins. We said these membrane associated proteins are long-lived proteins, and also long-lived proteins other than the membrane associated with proteins. The ATP-dependent degradation of proteins is in the cytosol for abnormal proteins, for example, cell structure is abnormal, or for proteins that are essentially short-lived, so they are normally have a short half-life. Those will be degraded by an ATP-dependent pathway, and this pathway requires ATP, so it requires energy, and it requires also ubiquitin. A new term here that we need to discuss is nitrogen balance. Nitrogen balance refers to the difference between total nitrogen intake and total nitrogen loss in feces, in urine, and in respiration. So the balance equals input minus output. And here is a graph that depicts time here in the x-axis, and it shows how degradation and synthesis are related to each other. So if we are synthesizing and at the same time there is degradation, at any point the difference between synthesis and degradation is the nitrogen balance. Nitrogen balance could be at equilibrium, that is to say input equals output, and this is the normal state in a normal adult. A nitrogen balance could be positive, that is to say input is more than the output, and this is the characteristic of growing infants and pregnancy, physiologically normal conditions in which nitrogen balance is positive. Nitrogen balance could also be negative, that is to say, the output here, the input is more than the output. The input is less than the output, and the output is more. So there is more degradation, for example, and more utilization, and this characterizes postoperative states and advanced pregnancy, malnutrition, as well as some types of chronic infections. Protein energy malnutrition, abbreviated as PEM. Protein energy malnutrition is a condition that is caused by deficiency of protein and calories. So it is related to protein and carbohydrate in the body. This leads to a range of health issues. And we have three types of protein energy malnutrition. Kwashiorkor, which is primarily due to protein deficiency, despite adequate caloric intake. So the caloric intake, the energy is not, the carbohydrates itself are available here. The condition is mainly deficiency of proteins. An example, famous example, is a child that is weaned into a starchy diet, which is poor in proteins. So starch here provides a carbohydrate, but the proteins are deficient in the diet. The second type of protein energy malnutrition is marasmus, which is due to severe deficiency of both calories and proteins. The third category is marasmic kwashiorkor, which is a combination of both. And this is a child with kwashiorkor versus a child with marasmus. Kwashiorkor is characterized by features that include edema, that is swelling, particularly in the legs and in the face. There is several skin lesions, fatty liver, hair discoloration, distended abdomen, and apathy. So the child will be apathetic and non-reactive. On the other hand, a marasmic child, as you can see, is wasted, particularly muscle wasting, with extreme weight loss, thin appearance, and dry skin, and general weakness. And a question for you here, from a biochemical point of view, marasmus is considered a successful starvation adaptation, while kwashiorkor is maladaptation. What do you think of this statement? I would like you to discuss it briefly. Our main topic for today is amino acid nitrogen catabolism. So we are going to deal with the amino nitrogen of the amino acid, and in the next lecture we will talk about how the body deals with the carbon skeleton. Catabolism of the amino acid nitrogen proceeds through four stages, including transamination, oxidative deamination, ammonia transport, and finally reactions of the urea cycle. So an amino acid will first undergo transamination, or if it is glutamate, it will directly go through the deamination. The ammonia will be removed, transported, and then it will proceed to the cycle. How how is ammonia removed from the amino acid? The first step in amino acid catabolism is the removal of ammonia, which is done by either transamination or deamination. Transamination is a reversible reaction. We have transaminases that are active in both catabolism and anabolism. We have certain exceptions here. For example, lysine, threonine, and proline, in addition to hydroxyproline, does not undergo transamination. Serine and threonine can be directly deaminated. The rest of the amino acid can undergo transamination. And transamination essentially involves interconversion of a pair of amino acids and a pair of keto acid. So this amino acid here, the amino group will be removed to form a keto acid, while the same amino group will be accepted by the other keto acid to form an amino acid. And the transaminases use, of course, vitamin B6 in the form of pyridoxal phosphate as the chromosomes. Transaminases or aminotransferases, the most important one here are alanine aminotransferase, abbreviated as ALT. It is also known as serum glutamate pyruvate transaminase, SGPT. We have also aspartate transaminase, AST, also known as serum glutamate oxaloacetate transaminase, SGOT. These are part of group of tests known as liver function tests. So when liver function test is requested, it usually include testing the enzymes ALT and AST. ALT or alanine transaminase, as the name implies, it includes alanine, and it is also known as serum glutamate pyruvate transaminase, SGPT. Why? Because here we interconvert glutamate with alanine. So glutamate with pyruvate plus pyruvate in the presence of the enzyme ALT or SGPT will give us alpha ketoglutarate and alanine. What happens here is that the amino group is reversibly, of course. Here is we are talking about this side here. The amino group is removed from glutamate, added to pyruvate. Pyruvate will come alanine, while glutamate will come alpha ketone. We said aminotransferases interconverts a pair of amino acids and a pair of keto acids. Here, the two amino acids are alanine and glutamate, and the two alpha keto acids are pyruvic acid and alpha ketoglutarate, and they are interconverted. It can be reversed in other direction, so the amino group can be removed from alanine, added to alpha ketoglutarate. Alanine will become pyruvate, and alpha ketoglutarate will become glutamate. Likewise, aspartate transaminase or serum glutamate oxaloacetate transaminase, because it interconverts glutamate and aspartate and their alpha keto acids respectively, oxaloacetate and alpha ketoglutarate. So the amino group can be reversibly removed from glutamate, added to oxaloacetate. Oxaloacetate will become aspartate, while glutamate will become, of course, alpha ketoglutarate, and vice versa. We have what we call key amino acids when we talk about the catabolism of amino nitrogen. These key amino acids act as a channel or a funnel for all amino groups from the rest of the amino acids. The key amino acids are alanine, aspartate, and glutamate, which are the three amino acids that we have just mentioned when we discussed the transaminases. Channeling into glutamate, all the amino acids will be transaminases will be channeled into glutamate. Alanine is also can be a substrate for glutamate transaminase. So by this, all the alpha amino acid nitrogens in all amino acids, of course, except the exceptions that we have already mentioned earlier, are going to be ultimately channeled into glutamate. All the amino nitrogens from the amino acids will be funneled into the key amino acid, and even aspartate and alanine amino groups will be funneled into glutamate. So glutamate is the ultimate receiver or acceptor of the amino nitrogen. And this is quite advantageous. Why? Because glutamate is the only mammalian amino acid that undergoes positive deamination at an appreciable rate. So glutamate undergoes positive deamination, which is important for removing amino group from the glutamate. Not just it is not just a subject or a substrate for the deamination. It does so at an appreciable rate. There is no other amino acid that can fulfill this criteria. So this is unique about glutamate, and that is why all the amino groups from the rest of the amino acid are funneled or collected into glutamate. A second form of, removing an amino group, the first one was through transamination, the second way is through deamination. Deamination refers to removal of the amino group without direct transfer to another compound. In transamination, we remove the amino group from the amino acid and we have given it to, or the transaminase enzyme will give it to another compound, which is alpha ketone acid, so it will not be free. While in deamination, ammonia amino group here is removed as a free without being transferred to another compound. Deamination can be carried out by glutamate dehydrogenase or L-amino acid oxidase, and the released ammonia will be readily used by hepatocyte for urea pyrolysis. And we will discuss why it is readily incorporated into urea as we approach the end of our discussion. Glutamate dehydrogenase releases free ammonia, releases amino group as free ammonia. It is unique in the sense that it can use either NAD or NADP as a coenzyme. It is allosterically regulated. Its activators include high energy signals such as ATP, GTP, and NADH, and it is allosterically inhibited by ADP. The reversal of the reaction is prevented by rapid removal of ammonia. So, glutamate dehydrogenase removes ammonia. The removes amino group of glutamate as ammonia, as free ammonia. Glutamate will be, of course, converted to alpha ketoglutarate. It is alpha ketone acid. It can use either NADP or NADP as a receptor of hydrogen, as a reductant. And this reaction can be reversed. However, continuous removal of ammonia from this site here ensures that the reaction goes in one direction towards the production of free ammonia and alpha ketoglutarate. The second enzyme that acts in deamination is the amino acid oxidase, which is a flavoprotein that catalyzes the deamination of an amino acids into alpha ketone acids. As a cofactor here is FAD, produces hydrogen peroxide and ammonia, and it has many roles in metabolism, microbial defense, as well as cell signaling. So this is amino acid that is going to undergo deamination by amino acid oxidase. It uses flavin, FAD, has a receptor of hydrogen, and this formation of hydrogen peroxide, which can be detoxified, of course, by a catalase into water. There is formation of an intermediary metabolite, that is alpha amino acid, which is spontaneously decomposes or is converted to alpha ketone acid with the release of free ammonia. This ammonia that is coming from either free or free, that's coming from deamination or incorporated with glutamate, has to be transported in the blood, either as free ammonia, just trace amount occurs in the blood as free ammonia. The majority will be within an amino acid that's are part of digestion, or from an amino acid that is in the blood, of course. In the tissues, ammonia is constantly produced by tissues and rapidly removed, of course, by our key amino acids, and finally incorporated into glutamine, and urea as a detoxification form. Liver is a final destination for ammonia. So once ammonia is removed from the amino acid, it is going to be transported to the liver for detoxification. What is the story of the link of ammonia and urea? Ammonia is a toxic substance. Urea is non-toxic substance. The body converts toxic ammonia, NH3, into urea, H4, which CO2, which is not toxic and easily excreted. So urea formation is a form of detoxification of ammonia. Urea as a form of detoxification has many advantages because urea is water soluble, neutral, and easily excreted by the kidney. And urea does not interrupt pH balance or cause immune toxicity. And urea is highly efficient as a detoxification detoxification molecule because it can carry two nitrogen atoms at one time. Ammonia itself has one nitrogen atom, but urea has two nitrogen atoms, and at the same time it is non-toxic and water soluble. That makes it an ideal a molecule for detoxification of the N-nitrogen of amino acids. Urea is a major end product of the catabolism of the N-nitrogen of amino acids in humans. For formation of 1 mole of urea, the requirements are 1 mole of alpha-amino nitrogen of aspartate, NH3, 1 mole of ammonium ion, NH4, 3 moles of ATP and carbon dioxide. Look here at the structure. The carbon here comes from carbon dioxide. We have two nitrogen atoms. One nitrogen atom comes from aspartate and the other nitrogen atom comes from ammonium ion. And the formation of urea is an active process that requires energy. For each 1 mole of urea, you need 3 ATPs. The body spends ATP here. That means that this is a significant process that um, necessitates energy production or any energy consumption. The urea cycle itself, as the name implies, it is a cycle, so it's a cyclic process without net gain or loss. It starts and finishes with ornithine, so ornithine is our initial substrate as well as the last product. It occurs partly in the matrix of the mitochondria and partly in the cytosol. And urea cycle, as we said, converts toxic ammonia to non-toxic urea. Urea cycle requires the action of 5 enzymes. The reactions themselves include formation of carbamoyl phosphate, which is a condensation reaction, and then formation of citrulline, formation of arginine-succinate, which is another condensation reaction, cleavage of arginine-succinate, and finally cleavage of arginine. So the first reaction, let's go to the figure here. So in the mitochondria, there is condensation of carbon molecule in the form of bicarbonate with ammonium ion is two ATPs to form carbamoyl phosphate, and the enzyme catalyzing this action is carbamoyl phosphate synthase 1. This enzyme here, carbamoyl phosphate synthase 1, is a rate limiting enzyme, so regulation of urea occurs at this level, at this enzyme. It is allosterically activated by N-acetyl glutamate. The second reaction is formation of the molecule citrulline. Citrulline formation occurs by transferring carbamoyl moiety from carbamoyl phosphate to ornithine to form citrulline. And the enzyme responsible for this reaction is L-ornithine transcarbamylase. We still within the mitochondrial matrix. And then citrulline exits to the cytosol where the rest of the reaction takes place. In the cytosol, or the third reaction in the cytosol is formation of arginine succinate. We said it is a condensation reaction between citrulline and aspartate by arginine succinate synthase. Aspartate here provides the second nitrogen of ammonia. The first, of course, nitrogen of ammonia came from the ammonium ion. ATP is also required for this second condensation reaction. The fourth reaction is cleaving argininosuccinate, yeah, to form arginine and to release fumarate. By argininosuccinate lies nitrogen is contained within arginine, and we know that arginine is an amino acid that contains two N nitrogens. It is a basic amino acid. And then arginine is cleaved by arginase to release urea and reform ornithine. Ornithine, of course, can go back inside the mitochondrial matrix to join again in another cycle of urea. That is why we said that it is a cycle that has no net gain or growth, and that it starts and ends with ornithine. We have certain disorders that are related to the urea cycle, and they are associated with each step, unfortunately. They are serious and potentially fatal, and they present with common clinical symptoms because all of them will more or less lead to abnormalities related to accumulation of ammonia, and we said ammonia is toxic to the body. The basis of therapy are almost the same, including limiting protein intake because protein here is a source of nitrogen, to remove excess ammonia and to replace missing intermediates and enzymes. We have, for example, deficiency of carbohydrate phosphate synthase 1 is known as hyperammonemia type 1. Deficiency of ornithine transcarbamylase is known as hyperammonemia type 2. In both of them, there is high levels of high concentration of ammonia in the blood. There is citrullinemia, argininosuccinic aciduria, and hyperargininemia. So we have 5 conditions that can be, or disorders that can be related to urea cycle. This is all. Thank you very much.